to where I am in my graduate program today and inspired a lot of the research that I do. So a little bit about Dr. Six. Her research focuses on the evolution and maintenance of symbioses, particularly in bark beetle systems and ambrosia beetle systems in relation to their fungi. Work in her lab highlights the influence of microfungi on greater ecosystem processes through different effects on their host insect, whether a bark beetle or ambrosia beetle. Her research also connects various aspects of bark beetle ecology and management, and also investigates how bark beetles may affect the ability of forests to adapt to climate change. And in addition to her position at the University of Montana, which is where she's joining us from today, she's also an extraordinary professor of ecology at the University of Pretoria in South Africa, and a proud member and fellow of the Royal Entomological Society. She also recently received the E.O. Wilson Biodiversity Technology Pioneer Award for pioneering research in bark beetle ecology and climate adaptation of forests. So her research is highly relevant to us today and very interdisciplinary. So there's something for all of us in her talk today. So today she'll be talking about why some trees survive bark beetle outbreaks, building a holistic answer to this question. Thank you so much for joining us. And without further ado, Dr. Diana Six.
All right, thank you. I really do want to thank everybody so much for inviting me to present this seminar. That is always one of my favorite things, is to connect with faculty and students at different universities. And so real honor to be asked. And it's always nice to be in person, but Zoom is also okay. I think a lot of us complain about Zoom, but I personally think it's a great tool and allows us to stay connected and to share science when we're sometimes in some challenging situations. So thanks for joining me. I'm going to kind of jump right in. And so, yeah, I'm going to talk today about looking at forests where bark beetles have gone through, developed these big outbreaks, and they've left some trees behind, and learning why some of these trees escape, and taking kind of a multi-trade approach to understand why these trees survive and maybe some of the characteristics we can exploit to help our forests kind of deal with some really challenging change. So I think most of you are familiar with the fact that we've just sort of come through one of the biggest, or probably the biggest, outbreak of bark beetles ever recorded. And that's the big mountain pine beetle outbreak. And if you look at this map, you see this huge red blob. That kind of shows the extent of the outbreak as of 2013. And of course, the outbreak extended for several years beyond that. So this blob should really be much larger. But it's really impressive, and it's kind of a harbinger of things to come. We're seeing more frequent, more severe, more broad outbreaks. And it's not just mountain pine beetle. There's been larger Ips outbreaks, spruce beetle. There's been huge outbreaks across Siberia, parts of Canada, parts of Europe. And so this is something our forests are facing in many, many places. And what I'm going to talk about today is kind of taking a less traditional approach to understanding what's happening for us and their capacity to adapt to things like bark beetles and climate change. And I'm going to argue that we really do have to take kind of some new approaches, that we have to move away from some of our traditional approaches a bit if we really want to understand the potential for our forests to adapt. Kind of the traditional approach has been to kind of focus on things like size class, how big or small are the trees, species composition, spacing, thinning, slope, aspect, soils. What are some of the attributes of these forests that may or may not influence bark beetle outbreaks and tree choice? But I would argue that there's been this huge gorilla in the room for a very long time, and that we have been really ignoring this gorilla, and that this gorilla is actually super important. And so what's this gorilla? Well, I would argue genetics. And the very reason for that, we know that kind of genetic diversity is really adaptation's toolkit. If you think about it, if you have a toolkit and you have a big problem show up and you've got two tools in it, what's the likelihood that you're going to have the right tools to deal with it? That likelihood is pretty slim. But if you had a toolkit that maybe had 25 tools in it, the likelihood that you'd have something at least is much better. And so if we think about conifers at least, which is mainly what I study, they are among some of the most genetically variable organisms that we have on this planet. And that means that conifers are really walking around with toolkits full of thousands of tools. Yet this is something that we pay very little attention to. And I think part of that is that people have the perspective that because trees are long-lived, that forests have a really low capacity to quickly adapt to change. And that turns out not to be the case. Many of the past studies and recent studies are showing that forests can adapt very rapidly, primarily when they have very highly selective agents and when those agents exert really strong selection pressure, which means killing a lot of trees. And so I would argue that bark beetles are exactly like that. They're very selective, and obviously, when they go through with an outbreak, they kill a lot of trees. Now, it's interesting that we seldom genetics because most of the factors that we usually consider important for survival, like drought tolerance, chemistry, growth rate, are all under really strong genetic control. So my feeling is that this is an approach that we can begin to take to get a better feeling of what's happening in our forests. Now, this is a picture that I use in one of my classes with some of my beginning undergrads to kind of introduce the idea of natural selection. And so what we have here is a really selective raptor. It preys on rodents. And you can see that we have these two types of rodents. There are the light colored and the dark colored. And because those light colored ones are more apparent, the raptor predates on those more heavily. And so over time, you have selection towards a higher frequency of these dark rodents. Now that's kind of the simple story. And there's usually a whole lot more going on, okay? It might be that not only are the dark rodents less visible to the raptor, but maybe they have behaviors that also help them avoid predation, or maybe they just taste bad or something. But there's likely to be a suite of traits that enhance their survival over those that are light. And so kind of with these things in mind, I have been asking kind of a whole suite of questions related to things that might be genetically based with these survivors. So first we need to know if they are genetically different. And if they are, that's great, because that means that whatever it is that's related to survival is likely heritable. And along those same lines, is survivorship related to tree defense chemistry? Is it related to growth rate? And is it related to climate sensitivity? And one of my bigger questions that all of this falls under is, do outbreaks accelerate or reduce, because it could go either direction, adaptation to forests, to both beetles and to a changing climate? Because we can see that a changing climate, particularly drier, warmer climate, increases susceptibility of forests to beetles. So I've been looking at these questions with mountain pine beetle. This is Dendroctonus ponderosi. And when people ask me, what does this beetle look like? I usually tell them kind of like a mouse turd. And so if you're looking at these hands, I think you'll agree with me that they do look a lot like a mouse turd. And actually the one on the left is a mouse turd. And so I think it's actually really remarkable that something this tiny, looks like a mouse turd, could have killed 70 million acres of trees over the last couple of decades. Now, obviously, a little beetle like this can't take down a big defensive tree all by itself. They use a pheromone-mediated mass attack that they use to overwhelm the tree. But before we even get to that point with these beetles, they have to actually choose or reject those trees. And those are the things that we're looking at. Why are some trees chosen and others pretty much completely left alone? So I'm going to talk mostly today about work that I've done in whitebark pine. This is Pinus albicaulis. We've done work in lodgepole and ponderous pine, but we're still adding sites and adding data. So I'm going to stick mainly to the species. It's what we call a naive host. It grows at much higher elevations than most pines, areas that have typically been too cold for the beetles. But with warming, the beetles have been able to move up into the tops of the mountains, and they've begun to attack these trees. And perhaps not surprisingly, these trees don't have much in the way of defense, and they really produce almost no resin. We have a hard time finding anything that even vaguely looks like induced defense. And so they just don't seem to have much in the way of defenses, unlike the lodgepole, which is downslope, that has a long coevolutionary history. Once they get into the high elevation, they can move through an area really fast. In the lower picture, you're looking at the high elevations in the Tetons. And this is what much of Teton National Park, lots of white bark across the west, Yellowstone looked like a few years ago. And the beetles just go through in like 3 years. It's like they don't even fight back. So I've been working in white bark pine, I think since 1999, which is quite a while. So I've been able to kind of watch the beetles move into high elevations and then develop these kind of devastating outbreaks and then collapse. And I found myself walking around in these forests, first kind of like, now what am I going to do? There's no beetles and no trees left. But then I started to look more closely, and even though these sites look really devastated, there were these trees here and there that were big, the right size, everything, but they were virtually untouched. And, I had friends say, oh, they just missed them by chance. And I just really never believed that, because when you have kind of billions of beetles flying around, they're running out of places to go, they're running out of food, they're not going into these trees, and they might be right next door to where these beetles emerged. And so I began to think, well, let's see if there's something different. So we located a couple of outbreak sites. One was Vipon Park, Montana, and that's the one you're looking at. This is actually one of the better parts of the stand. Most look much worse. This outbreak killed about 95% of the big mature trees. At Poverty Flat, our other site, This is in Idaho. It's about 250 kilometers away. It's about the same elevation, about the same kind of precip. The outbreak was about 70% mortality. And so we chose these sites to look at how these survivors might be different. And one of the things we chose these sites for is that they're nice and flat. And that's not just because I was tired of working on killer slopes, but it removes a lot of the microsite variation. So you don't have, the problems with aspect, slope, which is really a problem with dendrochronology, which we were doing. So we tried to remove as much of the moisture and aspect microsite variation so we could see what was just affecting trees. We also added this third site, Lacey Creek, which was only about 27 kilometers as the crow flies from Vipond Park, across a little valley. But it was a really strange place because even though the beetles really hit for us in this whole area, there were no beetles. Trees were just fine. So we wanted to see what was different about that site. So here's a very weird slide, but it's kind of my attempt to shove my entire experimental design onto one slide. So I'll go ahead and go through and explain this. At the outbreak sites, we located 3 different groups of trees. We have the bark beetle killed trees, which is B. We located a couple hundred trees at each of these sites, measured their diameter to determine what the diameter distribution was that the beetles were selecting at that site. Then we selected the S trees, which were survivors. And these are ones that had basically, well, they fell right into that same diameter distributions the beetles preferred, but they escaped. And then we had the G trees. These are general population. I could never really figure out a better name, but these were mature trees, but they escaped, we believe, because they were just a little too small. So they were just a few centimeters smaller in diameter than the trees that beetles were choosing. If you don't know much about bark beetles have a diameter distribution they like, and then they don't go into small trees. So we felt that this population would be a good proxy for what that population was like before the beetles showed up. So for example, at Vipond Park, where 95% were killed and 5% were survivors, we predicted or hypothesized that the general population would have about 5% genotypes that would match up to survivors and about 95% genotypes that wouldn't. So in other words, they would have probably susceptible genotypes. So for the bark beetle killed trees, all we could do is really take diameters and tree cores. So for the tree cores, we could look at how each tree responded to climatic variables in each year of its life, and also look for differences in growth rates. And then for the survivors, we were able to do a lot more. We took tree cores to do the same things. We measured resin flow, and I'm not going to talk about resin flow anymore in this talk, because out of the 80 or so trees that we punched for resin, these were all survivors. Only about 4 produced measurable resin. And so even the survivors just really aren't producing these constitutive defenses. We also collected from all of these needles, and from those we extracted DNA and then developed ISSR profiles. And ISSRs are inter-simple sequence repeats. And so they're the sequences that lie within microsatellite regions. And they're really good because they're super reproducible. They detect huge amounts of within tree variation, which is exactly what you're looking for a study like this. And also it's very cheap. It's a PCR-based method. And so you literally can do hundreds of trees, which is what we needed to do. Then we took punches through the bark and we measured phloem because that's been thought to be a selecting choice for beetles, that they like trees with thicker phloem. I'm not going to talk about that much anymore. It didn't seem to be all that important, but we also collected those phloem discs. And from that, we analyzed a number of terpenoids. And we did that, all those same things with the general population trees. So what did we find? Well, I'm going to talk mainly about our 2021 paper, which I sent out, I think the students read, in white bark pine, but we also did some of this with lodgepole pine that I'll talk about a little bit today. So for the genetics, this is the principal coordinates analysis using the ISSR data. And one of the first things we noticed was that within a site, comparing trees in a general or survivor categories, we often found more differentiation between those than you usually find between different populations of conifers. So there was a lot of differentiation between the generals and the survivors. We also noticed that there was quite a bit of difference between our populations, even those that were really close together. So like Vipon Park and Lacey Creek were only 27 kilometers apart, but they resolve quite different. So that indicates, at least with our populations, there's quite a bit of local adaptation. And some other work that people have done with some genetics with white bark are now starting to show that local adaptation can be really, really strong with this tree. But let me walk you through this principal components analysis real quick. Let's start with Poverty Flat, the Idaho site. The red circle indicates the survivors and the green are the generals. And you can see that those two groups of trees are really, really different. They have almost no overlap. And this didn't exactly fit our hypothesis because this particular population had 70% mortality, 30% survival. And we thought there would be maybe, 20 or 30% overlap. We just didn't see that. If we look at Vipon Park in Montana, the blue are the survivors and the yellow are the generals. And here we thought we would see very little overlap, but we actually have quite a bit. Although there is some pretty strong differentiation as well, but it's just not as extreme as we would have expected. The purple circle is Lacey Creek, where there were really no beetle-killed trees. Okay, there was like 2 in the whole area, but it certainly wasn't an outbreak. That particular population had some overlap with the Idaho trees, with some of the Vipon generals, but they were no overlap, at least with this analysis, with Vipon survivors. So there were differences, but not exactly in line with our hypotheses. This is a different way of looking at exactly the same data. This is Bayesian structure analysis, which looks for substructuring in particular groups of individuals. And at the top are the poverty flat trees. And that line in the middle separates the survivors and the generals. And you can see that they really split out pretty good, except for about 3 individuals in the survivors. And so again, at poverty flat, these 2 groups of trees are really, really different. At Vipon Park, the structure analysis indicated strong two groups, but there was a lot more admixture occurring between the different genotypes. And then at Lacey Creek, where we didn't have any beetle kill, we also have some indication that we have some substructuring. So there's quite a bit of diversity as far as substructuring in these populations. But something else we noticed was that when we looked at genetic diversity, we found some big surprises. So our hypothesis regarding genetic diversity was that in the survivors, because especially like at Vipond Park, where there was only about 5% of these big trees remaining, that we would see a big bottleneck effect because of all that mortality, that those few remaining survivors would have reduced genetic diversity because of just chance or drift. But we saw the opposite. It turns out that these abundant general populations, which are the smaller kind of replacement forests, had really low genetic diversity compared to those remaining survivors. We had to really think about that. And my best guess is that because they're smaller and younger, and we did the tree course, that they probably established under different conditions. And we know that kind of that early stage of establishment of seedlings and saplings is one of the strongest environmental filters that trees go through. And so perhaps they underwent some really strong selection during establishment that reduced their diversity. But that was a little disturbing because while the survivors tend to have, it looks like something it takes to survive that's genetic, it also indicates the replacement forest may be sort of without a very good toolkit. So this is my whole thing on lodgepole pine for this talk. We're still doing a lot of work with it. But at Vipond Park, where we looked at the white bark, it's sort of at a transition zone where there's quite a bit of lodgepole pine. It's right where it's moving into being pure white bark. And so once the bark beetles really wiped out a lot of the white bark pine and were kind of collapsing in white bark, they moved into some of the lodgepole at that same site. And they killed about 50% of the big lodgepole before they collapsed. And so we did kind of a smaller study, just doing the genetics to see if something was happening with the lodgepole as well. And so here's another structure analysis. And on one side, you see survivors. And unlike the white bark, it appears that there are two groups or genotype groups, the green and the blue, that accounts for survivorship. So there may be more than one kind of thing or group of things in these genotypes that account for survivorship. And then if we look at the generals, we see that about 50% are some other genotype and 50% have the same as the survivors. And this fits with our hypothesis really nice, that if you have 50% mortality, about 50% of those generals, if all of these were to grow up, would have survivor genotypes. So in this case, it actually fit. It's always nice when that happens. I'm going to move on now and cover a bit about chemistry. And these are cluster analyses. And what you're looking at with the heat maps are each line going vertically is an individual tree. And then the horizontal lines are particular terpenoids that we looked at. And the darker the terpenoid, the stronger the concentration, the lighter, the less, or the weaker the concentration. So let me walk you through the legend a little bit. The blue is for Lacey Creek, and those are all big trees in the population where there was no beetle kill. And then at Poverty Flat and Vipond Park, the light colors indicate the youngest trees. Those are the generals. And then, as you'll see when we move on to the tree cores, within the survivors, these big trees, it turns out there were older and younger trees. The younger, that's kind of a bad term probably. They were all old, but they were younger than the older ones. And so that gives you an idea of how to separate those out. So if we look at the top graph, this is with all the sites combined. And you can see that Vipond Park in the pink separates completely independently of Lacey Creek and Poverty. In that middle cluster, you have all those blue bars. Those are the Lacey Creek sites in Montana that are really close to the pink sites, but they come out separate. And then you can see that there's a cluster with mainly survivors for poverty. And then the next cluster are mainly generals for poverty. The lower graphs actually then are just for the two individual outbreak sites. So B, with all the pink, is for Vipond Park. And you can see that there's one cluster to the left that has all dark pink bars at the bottom. So this was a really distinct cluster. It was all old trees that survived. And then the generals and the old and younger ones were pretty intermixed evenly in the other two clusters. If we look at Poverty Flat in green, we see a bit more differentiation by survivor versus general. The big cluster on the left is almost all survivors, and then the cluster on the right is almost all generals. And so from these analyses, it appears that within a site, there does appear to be some chemical basis for survivorship, but there doesn't appear to be any single profile that is consistently associated with survival across the sites. Okay, and that was kind of a surprise. We were hoping to say, oh, well, if you have this profile, this helps these trees escape the beetle. That might be the case, but it only appears to be site specific. I really like this kind of an analysis because instead of comparing each individual terpenoid separately to see which ones are significantly different for each tree, you're looking at the entire profile together. And I think that with beetles, they read very complex signals, and that by looking at profiles, we're going to get a better feeling of how the beetles can choose trees. And actually, this analysis came out with a much stronger indication of preference than when we did individual terpenoids alone. Okay, let's talk about growth rates and some climate sensitivity in these trees. I just started doing tree cores about 5 or 6 years ago, and I totally fell in love. I don't know how many of you have done this, but just the act of taking a core out of a tree is just so amazing because you're really looking at like the tree's autobiography. You can see where it's had the rough spots, where it's had the good years, and everything in between. So you can really kind of see the tree's whole life. And so if you look at the cores here, you can see some big fat ones, those that the tree was really doing well. And then there's some skinny rings where things weren't quite so good. And so if we can figure out which ring goes with which year, we can then align that with a lot of climate variabilities, variables, and see how these trees respond. So here is a big graph, the kind I tell my students never to use in a talk, but I do it all the time. So it's not really fair. But I really felt it was kind of important to show this because it was something that kind of blew us away once we started looking at our tree ring data. What we found is that in each category, that trees had radically different growth rates. We were looking at trees that looked almost like the big trees were even age. They were big, most of them were pretty close in diameter, but trees that were standing right next to each other on flat ground, same diameter, one could be half the age of the other. So if you think you can age whitebark pine by their diameter, don't do it. It was really amazing. Two very different groups resolved at each site and in each category, indicating that there are subgroups that are genetically different with very different growth rates. So for example, if you look at poverty flat, the first two lines, older dead and younger dead, I'm going to get my cursor in here. Hopefully you can see it. The mean DBH was, they were a bit bigger, quite a bit bigger in the younger on average. But if you get over here and look at their age, these younger trees, even though they were much bigger, were much younger. And that was bore out by looking at their basal area increment, which is how much girth a tree puts on annually. Same thing if you looked at older survivors and younger survivors. The youngers were quite a bit bigger, but look at how much younger they were on average than these older survivors. So they were growing a lot faster. Same thing down here when you look at Vipond Park. These were virtually the same in diameter, but the younger trees were way younger. Same thing with the survivors. And then if you go to Lacey Creek, which these trees were a bit bigger than at the other two sites, but not a lot. It was shocking when we did the cores because their rings were so tiny. These are such slow growing trees. But again, you have this split into the really slow ones and then these slow younger ones, okay? So there's an indication we have variability in growth rates even within these categories. And when it's this distinct, you can pretty much assume it's genetic. Okay. These are mean growth rates for these various categories at the two outbreak sites. And I'll try and walk you through this kind of mess. I don't have the error bars in here because it became way too messy to even read. So I'll try and tell you what was going on. So if you look over here, you can see for poverty flat, you have this blue line, which are the, these are the ones that the beetles eventually killed. These gray ones are these older survivors that continue to survive. And while they look really close here, there was almost no variability in the two groups. So this was actually significant that those that survived were slower growing up to somewhere in the 30s, and these were faster. They converged through this period all the way up to here. This looks different, but most of this was actually not significantly different. But overall, they were slower growing, the ones that survived. If you look at the younger survivors and the younger beetle killed, again, those that survived were much slower growing in this case up until you get into the 70s, and then they coincided. And then if you look at the generals, they have really picked up and started growing fast. Given that we're seeing a pattern of slow growth related to survivorship, we're a little worried about these guys because if they're growing really fast in another 10 or 20 years, they'll be beetle bait. And we're really curious as to what this faster growth may mean. If we look down here, we again have kind of this difference. This was significant that the survivors grew a little faster way back, but then they grew slower all the way up until this period when they coincided again. And the same thing with the younger beetle killed and survivors. So overall, if you look at the entire growth period of these trees, those that have been slower growing, at least in their older age, and are the ones that seem to make it through. And again, here the generals are growing fast. I just threw this, no, I'm not ready for that one yet. Okay, so this is where I've just added in a graph including Palmer Drought Severity Index, which is a measure of drought. And so this is just for the last 50 some years or so. And with you see green, that means those years have been wetter than normal. And where you see yellow, they're drier than normal. And then these are growth rates again up here. You have those that end up surviving, those that were beetle killed, and then these generals. But what I have this in here for is mainly to show that there does appear to be a response to drought when it occurs in the growth. And particularly a drop here when we have this extended drought that occurred right before the outbreak, which started right here. Okay. So that was at Poverty Flat. And then we can see kind of the same thing again at Bipon Park, where we have lower precipitation, we have lower growth, and then with this extended drought here, we saw quite a drop in all of these trees. Interestingly, when we look at Lacey Creek, I've split these into the older and younger, but realize both of these are really old. It's just one's younger than the other. If we look way back, both of these groups grew very similar up until this point, where the youngers are starting to grow a little more. They're both responding a bit with greater growth, but overall they've remained really slow. And they show really very little variability, whether there's a drought or not. They seem really insensitive. Although right before the outbreak, they did both drop here, but there was no beetle response. Something really interesting about these trees is that they're so slow. For the other trees, their growth rates were up in this range, and these are way down here. And it just seems like the beetles just want nothing to do with these. So it does appear that slow growth rate may be something that's happening here. And Nick Keachus did a bunch of tree cores in Montana and found again the same thing, that slow growing white barks are just avoiding predation. So I'm going to do a real quick run through on the climate sensitivity. If you want to know more about this, you can go into the paper. But we wanted to know how these trees responded to temperature, precipitation, and drought. And so we did correlation analyses of basal area index by category, beetle killed, not killed, slow growing, fast growing at each site. And those were with mean basal area increment, mean growth rates for each year. And we found weak positive correlations for most months for PDSI, really weak. And we also looked at a 1-year lag to see if conditions the previous year affected the next year's growth. And we really found no stronger correlations, or very little. But this is not really the appropriate way to do these analysis for our questions. This is how it's usually done. But when you do a mean, it puts everything in together, and we're looking at subgroups that might be different, and we would lose that using means. So unfortunately, that meant that I had to go through and do hundreds and hundreds of correlational analyses of these for individual trees with something like 32 climate variables for each tree. Kind of glad I did, because as I did it, there were two groups that popped out really distinct. One group had correlation coefficients of greater than 0.2 with PDSI for 9 months or more each year. So they seem to be somewhat sensitive to drought. And then the second group which we call insensitive trees, showed little to no correlation with PDSI, just really didn't respond to drought. But about half of those showed positive correlations with March temperatures and negative correlations with December temperatures. So again, just like growth rates, we saw before that, some of them are growing twice as fast, and that appears to be genetically based. Here we have these two groups of sensitivity or insensitivity. I got very excited because I thought, oh, these drought sensitive ones are probably what the beetles are after. But unfortunately, that wasn't the case. These showed up in pretty good numbers in all categories. So it didn't account for beetle choice. So I'm going to wrap up now. I think I've gone on way too long. I just want to say that it does appear that trees that survive are genetically different. Terpenoids appear to affect choice of trees within populations, but we can't find any so far universal profile, at least with terpenoids, for survivorship across populations. Growth rate appears to be important with slower overall growth rates exhibited by survivors. Something I think is important is that these old trees appear to be repositories of genetic diversity, and that makes them super high importance because they're the toolkits. And so if they're not around to pollinate and have gene flow with the younger trees, this may be a problem. So we feel like assessments of genetic diversity need to start looking at multiple cohorts within population to get the full picture of what's going on. There's all sorts of conservation implications. If we plant, we have to realize we're probably planting with non-adapted genotypes. They may swamp out adaptation that's occurring. If we thin, we may increase growth. That may not be good with these. And as we're finding with lodgepole, trees make it better well. And salvage should be done carefully, at least in my area. When they go in and salvage lodgepole, the dead stuff, they take the green because it gets a better price at the mill and helps pay for the cut. I would argue we have to keep those on hand because they're what our forests need if they have the right genetics. With that, I'm going to quickly say thank you to Erica, Morgan, and Claire. People worked in my lab. I got a lot of this done. Dana Perkins, who's retired from BLM, but has been out in the field with me, helping me learn a lot of things. Peter Brown for help with tree rings, Amy Trowbridge and Michael Howe for help with the chemistry, and then these agencies for funding. Thank you.
Thank you, Diana. That was awesome. We have 10 minutes for questions, so I'll open the floor. Feel free to type your question in the chat box or unmute yourself to ask the question. We will also be continuing the discussion in the post-seminar discussion Zoom, and I'll post the link to that in the chat box while we do Q&A right now. So floor is open.
Hi, Diana. I have a question. That was a fantastic talk. Really wonderful. And the chemistry results were really interesting, where you mentioned that there's not a universal defended tree. And I'm wondering does that suggest sort of a local adaptation or attack preferences by mountain pine beetle within each of these areas?
I kind of doubt the beetles have the degree of local adaptation that the trees do. People have looked for that. They haven't found it, not in these populations. I'm becoming a heretic and thinking that, and Amy and I just stewed on this a bunch. We're starting to think terpenoids aren't important.
But you definitely saw those that survived were different.
Yeah, I mean, that they are for defense and some other things, but I'm not convinced they're the drivers necessarily of choice, or that they are part of the picture, but not enough. There's other stuff. How How do trees know if a tree, how do beetles know a tree's slow growing or fast? They're not out there with little increment cores, right? They don't have those.
Right.
So
Yeah.
they know
Yeah.
somehow.
And same thing with how do they know the size of the tree?
Yeah.
Visual.
You know, and they're
inability.
making choices. This is really interesting because this is not like some lodgepole forest where you have trees that were unsuccessfully attacked. They aren't attacking these trees. They're leaving them completely alone. So they might land, but they're certainly not going in. And I don't know. I sent off a whole bunch of samples to Katerina Villary for phenolics. It's not phenolics. You know, I don't know.
That makes it exciting. Pretty interesting and exciting.
No, it makes it difficult.
Yeah, I was gonna ask if you had put a sticky trap on trees as a mode of, are they landing or are they just actually not even flying by? But I'd assume they're probably landing and then just saying nope.
I assume that too, but you know, by the time we were looking, the populations collapsed.
Yeah.
And, you know, when beetle populations collapse, you can't find them. There could have been 5 billion the year before, and you can't get them into even a pheromone trap. So it's like, you know, I want to test this, but there's no beetles.
Yeah.
So yeah.
Thanks for your comments.
Yeah.
I kind of wanted to add on to that because, you know, I've kind of been wrestling with this whole terpenoid profile thing and does this matter, but in pinyon systems with IPS. With the mountain pine beetle though, do you think even, could it be affecting other aspects of this beetle, like the fungus and the ability of that fungus to establish in that host? Could terpenoid, you know, it's.
The fungi do fine. They don't have a problem with white bark at all. They're very similar to other pines in some ways. It's just they make less of some things and a bit of others, more of others. You know, I don't know. I mean, I was kind of flippant when I said rich that I don't think terpenoids are important. I think they're probably part of it, probably like just part of it. And we don't know what the other part is. You know, beetles have to pick trees for a lot of reasons. You know, we focus on defense because obviously they've got to get past defense. So they don't want to pick a really strong tree. But they also have to, that's just the defense is just the first few days or weeks of the invasion. They've got to be in that tree for the whole year. And they've got to deal with fitness effects of getting their young through. And so there's all these other things that are important, like food quality, nutrition, nitrogen, phosphorus amounts. And I would be surprised if they're not making some choices on quality of host as well, that's mixed in there. And we don't look at that. We only look at defense. So now I'm starting to think, how do you look at beetle choice for quality of food? Because they're having to make a really complicated decision that totally is going to affect their fitness. And they can't just only deal with fitness. So I don't know. I don't think I think I'm going to retire in a couple of years, and I'm not sure I can figure this out. But
we'll have to keep it going.
That's your job.
Any questions from other folks out there?
I'll throw in one thing. This is kind of a weird system because it's not, it being a naive host, right? You wouldn't expect co-evolved host profiles of chemistry. And you might think they've got just weird profiles that the beetles don't like. So I'm looking forward to looking at some of this in like lodgepole, where the terpenoids may come out as a much cleaner story, right? And that might be the case with nips too. Beetles that have been in a tree, and exerted pressure quite a bit over time. Unlike a lot of white bark stands that might have had a beetle outbreak or two, but that's it.
Right.
And some none. So it's sort of an odd system in a way. So that might account for some of our odd results.
I have a question.
Oh, look who's here.
Hi, Diana.
Hi.
It was really fun to hear about
Started in
this.
white bark with.
Yeah. So my favorite tree. I was wondering if if anyone's done a GWAS. It seems like that would be a really cool next step for this
It
kind of
would
work.
be.
You've got the right data set.
It would be. It'd be really nice. Are you going to do it?
No, but I can put you in touch with people who could.
Yeah, no, it would be a really good next step, especially since we can detect these trees with different traits. Would be super nice to do that.
Yeah, because then you could link maybe the Beetles plus the Drought and all of those other components. It would be really cool.
Exactly. Yeah, it'd be a great next step. And I hope somebody does it. I don't think it's going to be me. I know.
Understandable.
Well, no, I mean, I really am. Yeah, I've got 2 big projects and I'm also winding down on some of this stuff, but it would be really nice. I expect, because this tree is about to be listed as endangered. I think
Right.
there's going to be some money for this and somebody is going to jump on that. I hope. I know David Neal has now got the genome together, I believe, which will be really nice. But even there, you've got to know what is going on with your trees in order to know what genes to target. So, yeah.
Right. And I think Andrew Eckert, who did a postdoc with Dave, does a lot of GWAS, and that's who I work with on the Southwestern White Pine. So that's who
Cool.
I was thinking would be a good fit.
Yeah, hopefully he'll do it.
You should send him an email and put a.
Those of you that don't know, Kristen was my first graduate student, and it's all her fault that I started working in white bark, because that was not my intention, but that's what she wanted to do. And yeah, I've been up there ever since.
We could squeeze in one more question before our post-seminar discussion. Is there anyone out there who really wants to get their question in? Okay. We'll be meeting in the post-seminar discussion room. I believe Connor has put the link in the chat box. So I'll see you all there. And Diana, feel free to, take a short break before you log on. No rush. And I'll see you in the other Zoom room. You should have that in the email. Or it's also in the chat box.
Yeah.
Okay. Excellent.
Cool. Thank you, everybody.